my voice. Hello, welcome to office hours for Mystics and FlyWire. All right, yay! So we have three actually pretty good questions slash problems to kind of work with, and they all have to do with misalignments, which is actually like I'd say probably half of the most common errors you're going to deal with that the AI couldn't handle. And we can go from there. And you're definitely going to see that in the optic lobe, by the way, with more misalignments being the issue because of the way that the fly brain was imaged. The optic lobes, since they kind of, we'll just look real quick here at the mesh of the fly brain, since they kind of are on the edges here of the fly brain, they got a little detached during imaging and then kind of like re-sewn back together digitally. So that's why we have like those misalignments or jumps that might be happening, especially between certain areas. So let's jump into the first question, which was one that Black Score posted. Let me just turn off this brain mesh the other day. And it was about how to deal with some misalignments, especially since you can see where there's like a cutoff area that happens in the 3D here. So this is a case where we would, make sure to check the, the dendrite clusters or the axonal clusters and these things when you notice something really big like this where there's a break. So you can see here, if here I'll even just go into 3D full view where we have this misalignment gap here that's happening, and it kind of just cuts all the way across the top of all these clusters here. And just because it's so dramatic enough, we want to go through and look at all of those. So what I've done just to very quickly kind of easily, since I can see such great view here of all the problems, I'm actually just going to drop some annotation points. So I went ahead and did that just a little ahead of time. And I just went and created an annotation layer. And you can see I just dropped a bunch of hot pink ones where I saw that it was like a flat end that I should maybe check from this view. I'm just going to go back to my 2D view real quick. I want my clear one. Thank you. And we'll go through and just real quick check each of these points. And it's kind of nice because if you kind of drop these points here, then you don't have to precisely click. You can just, jump to that area where one is, and then we can just remove the annotation points. So we know we've checked that already. So here's the shift. You can see it because it goes to black and then comes back. The nice thing about this one is that we're gonna basically, it's only a couple slides and it's not dramatically moving. So we can kind of just think of it as we're closing our eyes for a long blink and then it's coming back. But the main thing I'm gonna look at is when we're in this, where it currently is there's this larger structure here with a bunch of these vesicles and then some larger intercellular structures inside of it. And that's a good way to know that, okay, I have my little horizontal oval here and it's going to be nestled in. It should be near this structure in another slide when I go to that jump. So I'm going to just scroll up real quick and I'm going to pretend to say like, okay, it's going to move probably along because you can see it's starting to move from the right to the left. So it should probably move further to the left. So I'm just going to do that and scroll up and I'm going to click on this piece because that seems to be the most logical. And that's going to add that on. And it looks like it's good. The other way we could do this is we could try selecting something near it. So we know that when this starts, it seems to be that there's this piece, this kind of L-shaped piece that's going to come along underneath it. Right here, we're seeing that start to happen underneath this green part here, even though it's not perfectly colored, but you can see if we turn off the color on the 2D, kind of where that's moving there. So, turn that back on. So I'm just going to select this real quick and see, all right, it's also having a similar cutoff. But if we add this, does that look like that? It's too big and messy, so definitely does not continue for this part. There's a lot of kind of clicking around that you do, and then going like that, this ends the structure. Based on this type of cell and what we're seeing over here where there isn't that cutoff, this looks good for adding on. So I'm really going to go ahead and keep that. I'm going to get rid of my little point here. I'm not going to quite merge it yet, just so I know that I definitely looked at it yet, even though I'm going to get rid of my point. So I'm just going to remove that real quick there. And another way to look at this is if we have like a continuation that we definitely know for sure that's good, say, you know, this little loop here, we can see that there's a shift, right? So anything that's added on this side will be shifted over a little bit more to the left, and this side will be shifted more to the right. So if we look over here where we added our continuation, this is more to the right and the shift is over more to the left, if that gives you kind of a clue on that. Let's see here. We'll do another couple of these. I think this one was a good example. Oh yeah, because this is a pretty big blob here. Similar where we're going to look at maybe the intercellular structures and how it's relating to it. So another way I could do this is by placing some annotation points, but I'm actually going to use another example, which is a little more clear than doing this one, because this is the pretty doable jump. So if we'll follow it along, that's where that continues. And the reason I know it is so I'm looking at some of the surrounding structures that are going on around this piece, and that the fact that it has a lot of intercellular structures in it. So yeah, obviously this is a little messed up, but we see this structure getting a little bit bigger from when it was before in this piece. And then when we move, we see that that's grown into being this, I think, mitochondria or endoplasmic reticulum, which is ER, kind of popping up here. And we also know it's good because we have these like 3 little nestled areas here. So if we go back to our green, we can see that we have these kind of main little parts kind of all coming around underneath it. The other thing too is we can look and we can say like, all right, is it relative that this larger mitochondria ER is up here is also up here compared to this. So there's a lot of kind of marking that you can do, and you can drop annotation points in different colors. Like maybe we could create another layer and be like, all right, you know, we know this guy is up here, and we know that in this one, it's this one where we started, and we could just kind of click between and see that they're relative to each other. And that kind of helps to see the picture of what's happening. So this big one here is shrinking when we go to the next slide. So I don't know, I don't particularly want to go through all of these. We can come back and go through a lot more of them if you want. But let me know if what you would like me to do, Black Score, for that. But that's a couple of those all added in. Nimi, yeah. So that's one way to use the annotations. It's kind of tricky because you want to make sure that you line them up next to each other. You can use a teleport. I think you can use the line annotation too to help kind of draw a line between the two of them. I'll have to do that in another video for demoing. I'm still figuring it out, and I want to make sure I have it pretty down pat before I show you guys for that annotation method. And as you can see, what I'm adding on here where these cut off, these are actually pretty, they're not just like this like little twig part. It's actually like the rest of the synapse point here, and we're adding in more branch structure. So it is important to go in and check all these and add them in rather than skipping over them or something like that. Now, if we looked at this one maybe way out here, this looks like it, I think it just ends. So yeah, there's nothing to add there, so we would just ignore this one. But it was good to check just because it was on that same kind of cut line that we saw there. But you can see really where that all goes across with the annotations, even though they're not placed perfectly in each of the spots. So I'm going to jump over to the one that I was going to show how to use a little bit more of that placing annotation points for jumping. This is an example that Ankri had. And at the end here, we have some pretty bad misalignment and shifts. And I'm just going to scroll real quick, and you can see how much we can look at this large cell body down here and just see how it moves. Like at one point, it goes down here, it comes up here, it's going all over the place as you scroll along. So how do you track that with your eye, especially when we're following something like this small little piece right here? The nice thing is that this small piece we know is related to this little almost face with these kind of like frog googly eyes. Make it a kind of thing. But these three neuron branches here are pretty distinct, and we know that it's nestled right underneath it. And we also know it's above this large cell body here, this soma. So that gives us some points of reference for where we want to go as we move along. And as we can see here, Ankri's already kind of followed along for where it is and gotten to this point. And then, we see that we're going to have some issues coming up because of the fold that's happening here in the EM. You can see those lines kind of going down. So what I'm going to do is, the other problem too is, I can't select anything in this slide in this area because it's so near the fold. The segmentation isn't there in the 3D, or it's just really sporadic and we don't know where it is. So I'm actually going to just add in some annotation points. So I've gone along and scrolled and just dropped annotation points where I see that little spot in relation to these three other branches here. I know it's kind of bouncy all over the place. And as you can see, I kind of was like, okay, there it is. And I see it's going across that fold. It's back here. We still have our little face to the right of it. And I've just dropped an annotation point where I want until I get to the very end here. And I scroll and I finally can see that I can select the next piece and add that in. And as you can see, Ankri's gone ahead and finished it because I'm using an out of date one. So yes, so that's where annotations, like, use them to draw on your 2D. It's a huge help, especially when you get to these things like this. So the other thing we can do is, let's say, we know, like this picture here, we know, oh, yep, these are the two in this, and we just didn't want to, we'll skip all the mess in between. What we could do is we could just, I'm just going to create a new, stop doing that, thank you, a new spot here, and I'll just make something where, all right, we began here, bring that up, control click, and then we ended here. And I could just teleport in between, and then I don't have as much of a shift issue, and it allows me to better see, okay, yep, that's the correct one. Now, let's say that we couldn't tell what was the correct one, but we know for a fact that maybe we have this distinctive one over here to help us, maybe pinpoint it. Let me just trash all these. Let's say we're following just this big giant cell body instead. So we'll scroll up until we think we're kind of in the safe area and go, oh, okay. Whoops. Scrolled out too much. All right, bring me back, please. Where are we? There we are. Say we know that we want to follow this large cell body at the bottom here. And so we could place 2 points and jump between the two of them. And that would allow us to maybe start, use that as the landmark that we jump in between on our slides in order to find the continuation. Oh, you guys were still relying on the old screenshot technique. Hey, yeah, I know, right? When we first started completing cells in FlyWire before the complete system, we would just take screenshots and use MS Paint on all the branches. So I completely understand. So yes, it's very nice to be able to just kind of draw in where you've been. And as you saw, that's what I was using too for, like, in a Black Scores example here, where I was using the annotation points to say, hey, I've looked here, or hey, I haven't looked here, let's look here next. So definitely use those things to your advantage for figuring this out. Okay, the next one I'm going to go to is one that Azure has submitted. And this one is, rather than a miscontinuation necessarily, it's just a slide that disappears. So I'm going to go ahead and I'm just going to hide slices. This one requires a little bit of human imagination. But it, and also, and a little less using annotation points for it. But Azure already gone through and checked a lot of the spots here on this. And here we can see that there's kind of a distinct cutoff because it's a little flat here. And none of the other spots seem to have any, areas of continuation. So if we look here too, we see that there's a black slide in between where we're missing one, and that's what's causing the issue for maybe why the computer couldn't see where to go. The problem is it's not very distinct where we're supposed to go, because the spot where it's continuing, well, what one is it? And where do we go from here? So basically what you're going to do is we're going to follow the flow of this point here. And we know that it's based on how it's growing. We started down here. It's growing up this direction towards the, not exactly towards the left and up, but kind of up around here. So what I'm going to do is, this looks like it could continue on. So let's just click it and see what happens. And that brings up this big old cell here that we definitely know our branch doesn't belong to. It doesn't look similar. We see the structure. It certainly doesn't go. But it does have this little nublet part here that I'm highlighting that seems to belong to what we want. So what we're going to do is we're going to select where that continuation might be. So let's see, we add this one in. I'm going to go to my segmentation layer here. And I'm actually going to make sure that I color anything I add into a different color from our starting piece. So we know that this one's bad, so we'll just make it red. I mean, that's the one we want. Whoops, we'll keep it yellow. We know that this one is bad, so I'm going to make it red. This is the one we just selected. And we selected it because it seemed to continue along this little sliver here. But when we look at it in the overall structure, that definitely doesn't fit with the structure of the rest of this kind of clump here. It comes out way too wide. And anyways, if we look in 3D, this is kind of going up this way, and this feels like it should be going across to the right. So let's continuing just adding some stuff by elimination. So I'm going to turn this other blue piece red. I know it doesn't belong, but I've added it in. So let's connect. Let's see what's going on with this neighbor piece here. Well, that looks possible. That could be good. This pink one. Let's color it, green. Killing you with stoplight colors here. So, all right, that maybe could continue, but then we have this where it's next to the piece that we think it moves into. So that's not quite perfect. Is there anything else near it that might work? Well, let's see here. We're gonna kind of scroll here and we're gonna hover over where we know this is the little nub part that we see here that I keep highlighting with my cursor, this little crescent piece. So I'm gonna go into the black slide here and see like, oh, maybe I should click this. Well, that's interesting. It goes down this way. Let's color it blue. And we can see that there's a little jaggedness here. So let's get rid of our red pieces. Let's compare. Do we like this green piece? Well, this green piece, if we look at it and follow it along, it also has some issues, but it kind of just looks like a twig and we want it to continue. And that's just going into more twig here. And this seems to end here. So this isn't feeling quite right, this blue piece. I mean, this green piece. So I'm going to keep this blue piece right here. And just kind of by a process of elimination, I'm going to look out and see how that goes. I'm going to just turn off your annotation layer real quick here. And that looks pretty good. let's see here. So we have our cell as a whole. Looks like we have our main backbone coming up here. So the branch is flowing with the rest of the cell. It's kind of, flowing out like a starburst here. And if we compare it maybe to some of these other ones, well, it has a similar bouton with a long strand in between and maybe a couple little twigs coming off. We see that in another neighboring branch over here that looks like this is a little longer. So maybe this is actually where it belongs, and I have a feeling it is. And we can kind of see in the 3D that it connects here. The other thing we can do is let's bring that back, that red crescent shaped one. Is if we look at this in 3D and even in 2D, it kind of fills in a little gap here. So maybe that is where it goes. And what we can do is we can try to see if maybe we can find a back trace for the blue piece, just like we do in FlyWire. And well, if we click this, well, that's a huge glial mess that fills that all in, and that certainly doesn't have any merge like pieces that are merged onto the glial mess. That look promising. And any other continuations? Well, let's see what this. No, that goes to this little tiny piece here. So that seems to be different. So it's a little bit of trial and error, but it seems to flow very well. And the fact that it fits with the rest of the cell tells me that that's definitely where your continuation is on this. Notice I didn't use any annotations really for this. I mostly just used it by coloring the segments different colors, whether or not I decided that that could be a possibility that it belongs, or that it couldn't be a possibility that it belongs. And you kind of have to look too, like this area here, it looks like, we have some squeezing going on with these long strands here. And that certainly seems to be the case, that something came in, this guy, our red piece, our infamous red piece came in and kind of maybe squeezed out a little bit of the strand here. So it got very thin. And then because of the black slice, it's hard to find the continuation. All right. So that's a couple of kind of how to handle tricky traces kind of situation. And once again, I noticed here Azure J was using the annotations here to make notes of where you've been and where you might have a possibility. So excellent, use that to your advantage for doing this, especially on these pieces like this. The example that Ankri gave, she was having trouble with finding where this thing went. And she used a bunch of annotation points to say, oh, I looked at this, these all don't work. And then finally found this little tiny connection over here where it fits in with the rest of the cell. And we know it does because it's similar kind of structure with the kind of like the fine hairy kind of filaments coming off from these thicker bouton kind of, and also it kind of helps to wrap around the central main backbone here, similar like its friend up top. So that kind of is one way to look at it. So yeah, all right. Any other questions, or if we want, we can go back to doing a little bit more tracing, or I will have a look to make sure we haven't missed anything else. Anyone else has posted anywhere? I think we're good. I can go find more of your twigs ending here if you want. Flag score for you, and then I can send you the link so we don't do double work, but whatever you would like. But yeah, a great way is just to look and see like, okay, that finalizes that out for these things. So I'll get rid of this one. Let's see here, what layer are we on? Oh, we don't care about that layer. There we go. And that highlights that one right there, and just delete that. So we'll definitely make some short little videos, I think, for using some of these more annotation point kind of heavy things, especially in regards to misalignment jumps. and kind of just following the flow or the vector of the neuron branch as it goes. What's really helpful though is, yes, I agree with you. Yeah, especially because you get so much jumbling going on with these, like, like, oh, what's going on here, compared to what's going on here. Um, I think of it as if you've ever done any animation or anything like that, you do keyframes where you want the main, action to happen, and then the computer will fill in the filler movement between, moving your arm from an up position to a down position or something like that. Is kind of what we're doing with those annotation points. So it's a great way to kind of follow that shift. And I know that that's one reason why I want to show how to use the line one. I need to talk to a tracer that's at Princeton, in our Princeton lab, who has dealt with a very large shift, and they've just basically gone and done a bunch of little lines, and it really kind of shows the shift diagonally across, so they can more easily follow like a big bundle of like a bunch of these little ones all across the jump. So yeah, we'll make a little bit more little maybe snapshot some of these videos for how to do it. The other thing too is, you've noticed I definitely, and I know you guys too, that's partially why we are trying to post up the different cell types. The nice thing is the optic lobe, they're very distinct cell types, all within that a single column, and we'll see them in each column as we go along. So we'll get pretty familiar with what these cells should look like, but it's very handy that we already have the overall structure of the cell built out. So it's a lot easier to see where there is a merger, like, this one's definitely got a merger here, and this is a merger down here. And where a branch might just, it doesn't look like the rest of the cell in terms of its growth. Even if it's something even as sparse as this, maybe we can see that this belongs with the rest. All right. I think that's it for what I was going to show for little tips and tricks today, because it was mostly just about the annotation jumps. And as we saw, Ankri had a really great one here with the big dramatic one. And this will happen commonly near, as you can see, the what's happening is it's going from the, I believe this is, let me bring up my 3D. Yeah, so it's going from the optic lobe into the central brain here. And so there's a bit of a jump between the two of them, like a chasm that goes between them. And so that's why we have this kind of issue around the edge. Yeah. I'm glad that those have been working out. I went through and I very unscientifically named them just based on what they look like. But it ended up that they're actually really good groupings. And I think because, we're visually inspecting them, having a visual name for them kind of helps. Yeah. Yeah. The annotation tools, as you can see, I'm very comfortable with using points. I'm still figuring out the rest of these little ones here in terms of what you can use them for. And I want to make sure I have those mastered enough to make a little video for you guys on those. I think Ankri on hers, I remember, she somehow used the box one. And I thought that was very cool. Yes. Great. Having annotation videos, yes, little snapshots will be good for these kind of things. Yeah, because then you could circle, I guess you could circle the box over this kind of whole area, so This is actually the one thing that'll happen with these types of cells like this, is I've seen several of them that all have this kind of cutoff like this. So be sure to kind of keep an eye out for it when you have these. They're not like this one's, it's not immediately distinct that there's that cutoff until you get to the right angle. for these. And that's why, we do a quick, we're not going into the weeds per se for looking at this, but we're looking at it to double check that there isn't anything kind of obvious and cut off in these clusters of branches. But we're not worrying about adding all the little twigs themselves on it. All right, well, thank you guys for joining. I will be around for a little while longer, and I don't want to take up too much of your time if you need to pop out. Yeah, yeah. So black square, we're still, that's why I'm using all these examples of where it's you want to add and where you don't want to add for twigs, because it is a little bit objective or subjective, I should say. But don't sweat it if you miss some, when you have completed a cell. I'm sure I have over time. But at the same time, if you see something where it is a larger one with like a synapse point in it, like this would definitely be one that we would try to make sure we didn't miss. This one, I'd say if you maybe missed from annotation yet. I'd say if you maybe like didn't add on from this annotation point to the right, like this Y shape, and you missed adding that, it wouldn't be the a huge issue. That's something that the computer, the AI we're training we're hoping would be able to add on if that got missed. But in general, this is big enough that we want to make sure we add it. I know that that's something that even, how to know if a cell is complete is, those instructions on, is the twig done? Do I need to add more? Do I not? And I think what's helpful is that we have smaller kind of clusters or tiled clusters on these types of cells that we have in the optic lobes. So we don't have to worry about it as much as some of the ones that that you probably saw when you went into default mode in the sandbox. Those cells tend to have much bigger dendritic clusters, or even just whole kind of canopies or umbrellas on each side. So I think that's a great strategy right now to do AzurJ. That's pretty much what I do myself. Is I spot check it, and a lot of times I'm just looking to see is it a long skinny one, then I should look at the end of it probably. If it's a short skinny one like this one right here, I wouldn't look at the end of it, because it's probably doesn't have anything more to add to it. If it's thick and short like this, definitely we would want to check the end of it. Yeah, we'll go through. I think Amy has a list of these cells that you guys have done so far.
Yes, Sven made one yesterday. I can ask him to make one for. So he made one the first day. I don't think he made one yesterday, but let me ping him and see if I can get that.
Yeah, that'd be great. Yeah, because that's one of my to-do lists today, is to go over those cells just to double check for you guys, since we're still first starting out. And certainly we can go back and find that cell for you, black score. All right. Yeah, I know. I wish we had a list. I think that's something that we are even working on for in your little admin panel, is not even just the number of cells you've done, but maybe eventually getting a little cell history would be really nice. So you can go back and look at everything.
Yeah.
I think it'll be helpful too if you are like, oh, that's like that one I did like 5 cells ago. Let me look at it and see what that structure looked like compared to the one that I have now. That's certainly very helpful. So, and if you are feeling very ambitious, great. Thanks for joining us, Jay. We'll be here next Thursday. And as always, just ask us. And I was saying, if you're feeling really ambitious, you can always, after completing a cell, open up a blank, a FlyWire, and then you can add multiple cells to the list. So we could just, if we wanted to, we're like, oh, I completed this one. Let me you just hit this little clipboard, and that'll copy the segment ID, and you could just, pop it in, and then you could keep a little NeuroGlancer link for yourself that you just add to of all the cells that you've completed over time, if you desire. So that's just, but that's your own house note keeping that you'll have to do, and we don't want to have that. You guys have to do that, but just if you're feeling like it. All right, I think we'll wrap up. If you don't have any more questions, Black Score, unless you want me to trace something else or keep tracing another cell that I've been working on or something like that. Otherwise, thank you so much for coming.
Thanks, guys. I guess we'll do this on Twitch next week.
Yeah, we're going to see if we can figure out how to do this on Twitch. So then that way folks can more easily access these. So yeah. All right. Oh,
Hungry, hey.
Hungry, hey, we were just wrapping up, but come on in. We're still here. You're having trouble with annotations being visible. Are you saying when you zoom out really far or when you're zoomed in? Is it because you've placed the point in the 2D? Let me think about this.
I don't see them at all.
When you place them?
I place them and I get the ID, but I don't see them in the 3D or 2D.
Okay. Let's see here. What's what I'll do is let me delete this annotation, and we'll just do a quick see if process of how I'm doing the annotations, and maybe you can see if maybe you're doing something different, or maybe going from there. So we're gonna right click or control click to get the layer, and then remember, if you left click the layer, it'll cross it out so you can't see it anymore. It's not visible. Also, if you hit your numbers on your keyboard, it will turn that layer off. So right now I've hit number 2, which is this layer number 2, and so we can't see any of the segmentation. But if I hit 2, it'll come back again. So that's just kind of a basic, if you accidentally hit something or left click on a layer, just double check that. For the annotations, let's say, I'm looking at this if I want to place points where I see a bunch of cutoff things that are all gotten sliced off here. I'm going to go ahead and I'm going to add them in hot pink. And I'm going to control click, and I'm wondering, so at some point, like at some angles, like right here, like even though I know that annotation point is in there, I can't see it. So it might be that you need to zoom out. It should show. You see, further you zoom out, it should show. It may not be visible when you're up close, but the best way to maybe center yourself on it in the 2D, right, is to click on it, and then you can see it at least in the 2D and know that it's somewhere in the 3D. That's probably because what's happened is, since we placed the annotation point in the middle of the 3D structure, it's hidden in there. I think
It looks like I have the left clicked on the annotation layer or something. It's
is it crossed out?
Yeah, cross out.
Yep. So just left click it again and it'll pop back up.
Hmm.
Should work.
When you have several different cells or in there, is there any way to click on the numbers and you get centered on that cell if it's a small nub?
Mhm.
I don't think there is a way to do that
I
currently.
don't think so.
That would be a really nice feature though.
Yeah, yeah. Unfortunately not. Yeah, it makes it a little difficult for jumping like that. So, wondering if we can. Just the left click solve your problem of not being able to see it, and then just the fact that sometimes if you zoom in real close, if it's inside, it scales with your zoom, so that's why it shows more when you're zoomed out when we have the big pink dot, but then when we go in, it gets really tiny and we can't see it within the 3D like this. Unfortunately, there's not much we can do about that problem in terms of how that works. So. Do you have any other questions? I used your, oh, let me get rid of this one segment here. Just remove that. I used your example from yesterday for today because you did a great job of finding the cell body, by the way, on this cell. So that is great. And what I did, I noticed you used the box here for your annotation. I also dropped a bunch of points where I like checked the ends real quick to say that I had looked at them when I went through to just double check that this was indeed the only good connection for this cell. So good job on finding that. These are I think this type of situation where you start with like one of these clusters like this, where there's no obvious like continuation spot, they are one of the hardest things to do. So great job catching that. So that's where I guess our FlyWire experience of going through cube by cube, slice by slice really comes in handy for this. Any other questions? You can type in chat if you don't want to speak. And if there's anything you want me to go over, I can talk about how we're using annotations a little bit more for jumping. I think I gave you used the annotation points, I'm assuming, for leaving, how I sent you the example of leaving a little kind of breadcrumbs, since we can't select any segments here. That's not highlighting anything until later. Then we can add them in.
Yeah.
So the other thing is that what we can use is, especially in your case here for this one, I'm gonna just, let me see, I'm gonna turn off that annotation. No, not this one. This one I wanted is, let's say that we have a large structure near where we were working that's pretty identifiable, and then we have all the junk in between that's hard to see what's going on. But maybe we scroll up and we're like, okay, this is where this large cell body is. We can place a point here, and we'll place a point in this soma here that we know is the same thing. And then you can kind of use the annotation to jump between the slides and then more easily find where the piece that you might be following is relative to it. And then that way you're not scrolling through, 12 or more slides of a whole bunch of stuff that your eye can't easily track. And see here, we can just kind of more quickly see by toggling between these two. Just an example, but in your case for this one, because we had these three very easily recognizable spots here, it was pretty easy to just kind of drop the breadcrumbs like we did here.
Okay.
Yeah. So feel free to use the annotation points. No, you already kind of used them a little bit here, but drop them where you need them. And what I've done is you can see, I just added a few more layers so I can change the color if I know that that's where something is that I'm following. So.
I don't have had a lot of time to try it out this week, so
No worries. So
I'm probably have more questions later on.
that's what we're here for. We're going to be having these every Thursday for a while. So feel free to pop in with the questions or send us them or post them in the discussion board, and we'll try our best to get back to you as fast as we can. So thank you for popping in. So any Amy, go ahead.
Oh no, I was just gonna say that when we were going over the edit suggestions, this girl Claire, who's been she's a postdoc and she's spent 10 years working on Fly and has been working with all the scientists on FlyWire. She literally kept calling everyone's edits beautiful. Like she was like, this is a beautiful edit. This is just so beautifully done. And she was really impressed with the attention to detail and the skills that you guys showed in those submissions. I don't know if it came across in the feedback, but it was very, you guys really impressed the scientists.
Yeah, no. So that's something that we're very happy with because you guys are all a great community of folks. So. All right. Well, it's getting on to be 2 o'clock and I don't want to keep anyone for too long. So if that's it for all our questions, we'll go ahead and wrap up. And we've been recording this session. We're gonna
We'll put it on YouTube and send it out maybe in the forum, or we can just email it to you. And then the future ones, well, it should be automatically archived on Twitch.
I hope.
We'll try it out. We're learning.
Different situation.
I have not tried Twitch before, so that will be a new experience.
You can even just watch them, so that's handy.
Yeah, I think it's like YouTube. You just click on it and then you can watch a video and there's like a live chat with it.
Yeah.
Well, thank you guys so much for joining. And thanks for joining FlyWire. Woo!
Yeah, thank you. Yeah, we're excited to have you guys jump in on this new data set. It's been a crazy ride to get here. And as you can see, the software is a bit interesting. So we're trying to make it a bit more friendly for you all and for ourselves too.
Yeah.
Okay.
See you guys in FlyWire chat and maybe here next week.
Yes, exactly. So happy tracing. Hopefully you're having wonderful weather wherever you are. And we'll see you guys online.
Bye.
Bye.
Bye.